Hello everyone, my name is Aarón Espinosa. I'm a graduate student at Simvesta Virapuato, and I'm going to talk to you about microbiome diversity of spontaneous fermentation in Mexican agave distillates. Fermentation is a process guided by microorganisms, let's say bacteria, yeast, and fungi. These microbes take advantage from complex sugars provided by agave bagasso and juice. Each agave species slightly changes its sugar composition, and at the same time, each microorganisms provide different secondary metabolites which will give aroma and texture to the final beverage. Nevertheless, there is always a competition for surviving. This biochemical fight outcomes in a dynamic of microbial abundance along fermentation. And here I want to make sure it's clear that microbial abundances are not static through fermentation but changing, and according to that, it is the metabolite production in the media. Molecular markers to assign taxonomy has been widely used together with new generation sequencing. In the case of bacteria, the ribosomal gene 16S is used. It has a length of approximately 1400 base pairs, and among its sequence includes 9 hypervariable regions. These are employed as markers to elucidate taxonomy. For yeast and fungi, we use the ITS1 and ITS2 regions, which are flanked by ribosomal units 18S and 26S. The ITS2 region is well known for its length plasticity, ranging from 190 to 500 base pairs. For both 16S rRNA and ITS amplification, specific primers are employed depending on the interest region. For example, B3, B4 for 16S and ITS2 for ITS region. After sequencing and quality control processing, we can manage data into specialized software like Data2, which infers applicant sequence variants or ASVs, representing each taxon. This software relies on databases as a double-check strategy and additionally forewarn from technical common errors like PCR for a subsequent analysis. There are many available tools for meta-barcoding data management. One of the populars is Phylosec, which generates an object that serves as a container for taxonomy, ASVs, abundances, and metadata. And this is very helpful when measuring different diversity indexes, statistical analysis, and plotting. This work includes fermentation samples from 5 different states in Mexico, which are colored in this map, plus also 5 agave species. We then seek to predict main diversity and structure community drivers in mezcal fermentation. Shannon index shows diversity in each group. The agave species with high bacterial diversity are agave Karwinskii from Oaxaca and agave rhodacantha from Jalisco. Instead, for yeast and fungi, the agave angustifolia sampled in Sonora deploys the highest fungal diversity. However, the agave angustifolia with agave salmiana from Jalisco has the highest Shannon diversity score in the whole analysis. These plots here are called NMDS, which means non-metric multidimensional scaling, and their major objective is to represent data using a reduced number of dimensions for an easy visualization. In this case, employs a similarity matrix and then give this plot which indicates significant differentiation levels among groups. We then identify 4 differentiated groups in this study. Agave angustifolia from Oaxaca in brown solid square, agave angustifolia from Sonora in brown crossed square, the mix of angustifolia and salmiana from Jalisco in solid green triangle, and interestingly agave potatorum from Oaxaca in solid blue square. We found exactly the same clustering pattern for yeast analysis, demonstrating microbial community structure through this study. These heat maps show the relative abundance of 20 main genera of both bacterial and yeast fungi communities. For bacteria, Lactobacillus dominates through the whole fermentation process, while leuconostoc shown to be more abundant in regions like Guanajuato and Sonora. The microbial core in the fermentation is represented by the first 5 rows in the left figure, maybe excluding only Oenococcus. On the other hand, for yeast, Pichia and Saccharomyces shown to be the most dominant genera through the fermentation process. Meanwhile, the microbial core are the first 4 rows in the heat map. Finally, the main conclusions in this analysis are that we found a correlation between agave species and geographic region with the microbial community diversity and structure. Also that Sonora is the most diverse geographic region in fungal genera. We were expecting also that Lactobacillus were the most prominent genus in the analysis. Agave rhodacantha and Agave Karwinskii are the species with the most diverse bacterial genera, and on the other hand, the mix of Agave salmiana and Agave angustifolia have the most diverse fungal in genera. And also that Pichia and Saccharomyces have similar abundances across sites and agave species. And I want to thank all the colleagues, producers, and the Yeast MX teams for all the effort. Thank you so much.